Alright, now that we've made it out of the filtration membrane, we are now officially filtrate. We've got filtrate now, not urine yet, filtrate. And that filtrate's going to make its way to the proximal convoluted tubule. Now if you zoom in here, if you are confused about what gets reabsorbed and secreted here, these images do contain that content. So sodium, we're going to have glucose, amino acids, phosphates. That means, do you guys know what an amino acid is? What's an amino acid? Yeah, it's the building block for protein. So when we say amino acid, we mean protein subunits. It's the same thing. Glucose is a sugar subunit. Then phosphates, some chloride, some water gets reabsorbed here. And again, I'm telling you, in addition to this, that drugs will be secreted here at the proximal convoluted tubule. Histologists love the kidneys. That's kind of the favorite for histology because it's always very, very recognizable. You can always spot a glomerulus within a histology sample. They look like this. There's this little tuft of capillary. A lot of those cell bodies in there are podocyte cell bodies. That's why it's got those dark purple dots. And then you have that capsular space right there. So it's just really fun. And I was telling you about how the PCT has lots of tall microvilli, right? And that increases surface area so that we can have a lot of reabsorption at the proximal convoluted tubule. And that's actually something that you can see on the histology as well. You can see the microvilli in these tubules. So you can see the tubules around the glomerulus in histology. So you can see that luminal space has all these little finger-like projections in it, and that's how you know it's proximal convoluted tubule. As for distal convoluted tubule, it does not have those long microvilli. It has short, sparse microvilli, if any, so that distal convoluted tubule is going to look relatively clear in comparison. That lumen space is not going to have any finger-like projections in them. Not to say that there's going to be a detailed histology image for you to label. I'm just pointing out that connection between structure and function, those microvilli giving you absorptive surface area, does give you that significance of the PTC being the primary site of reabsorption. And you can even see that at the physical level. Okay, nephron loop, descending limb is going to reabsorb H2O. Ascending limb, we're going to focus on that sodium, but it also causes reabsorption of chloride and potassium. Now this is a calculation of osmolality, which is concentration. I'm never really going to have you learn these numbers. Don't really worry too much about the specific numbers. There is a series of videos that we're going to want to watch today, and it's called Biomed Sessions with Ruth. And I wish she had done more videos. She only did the renal system, but she did the renal system very, very, very well. So we will link to those. I think on our modules they may already be linked. And she's really easy to find on Google as well. She explains the concentration gradients and the countercurrent mechanisms and everything of how this creates the osmolality throughout the renal medulla here. Better than I ever could, frankly. But again, I'm not going to actually make you do any math. All you really need to know is that we make a salty medulla, and as such, water comes out. That's really all there is to it. And because we lose water here and salt here, by the time we get from the PCT to the DCT, we've concentrated that urine quite a bit. Now again, thin descending limb is permeable to water but not to salt. Thick ascending limb is impermeable to water, but there are pumps for the sodium and the chloride. Because the sodium and chloride is pumped out in the ascending limb, the water will be passively reabsorbed. And again, that's happening across the tissue. So this happens out into the renal medulla. But ultimately it gets reabsorbed to the cardiovascular system. The cardiovascular system around the loop of Henle is known as vasa recta. And again, that gradient, that saltiness gradient set up by that ascending limb is utilized at the collecting duct. In the presence of ADH, Aquaporins will be added to the epithelium here, and water will be passively reabsorbed. So now our urine has made its way to the distal convoluted tubule. It's going to secrete potassium and hydrogen ions from capillaries into tubular fluid. What's hydrogen ion associated with? What do you think of when you think of hydrogen ions? Acids, yes. We mentioned earlier that this is going to help us with our acid-base balance. And this is one way that it helps with acid-base balance. We can literally pee acid out. And that is going to, we will have slightly acidic urine. And that's actually going to help too. Having acidic urine is going to help prevent UTIs. So yay acidic urine. And also yay acid-base balance. So that's just one mechanism by which the kidneys are helping you with acid-base balance. And that DCT responds to aldosterone. And in the presence of aldosterone, it will retain sodium, reabsorb sodium, therefore reabsorb water, but secrete potassium. Okay, I think we're pretty good there. Now, fun fact, alcohol is an ADH inhibitor. Alcohol inhibits the actions of antidiuretic hormone, thereby having a diuretic effect. And that's why you pee a lot when you're drinking a lot. And it's not just the literal quantity that you're drinking. It is also the diuretic effect of alcohol working against ADH. A lot of drugs are going to work on this system. You're going to have sodium reabsorption inhibitors. If you can't reabsorb sodium, you can't set up that saltiness gradient. Loop diuretics are going to impact the loop of Henle. And osmotic diuretics are going to be things that won't be reabsorbed, so water remains in the urine. So high glucose of a diabetic patient means that there's lots of stuff in the urine, and then water stays in the urine, and that gets flushed out a lot. Yes?
It made it sound like ADH, like when you drink alcohol, like it's bad that it's the ADH is being inhibited.
In a way, it's going to dehydrate you. The more you pee out, the more dehydrated you are.
But isn't it like a drink or 2 drinks per guy, or 1 per like a day? Like, isn't that supposed to be like healthy?
Kind of. We could talk about that if you want. It's kind of the way science is. You know, if you turn on the news at night and it says, chocolate causes cancer, chocolate cures cancer. It's kind of one of those things. There was a study a while back that said something like a certain amount of alcohol basically prevents you from dying of other things, was the argument they were making. But there's more recent studies are kind of saying, no, it's not really healthy or good. It's just wasn't a very good experimental design. Like, well, you're going to die of alcoholism, but you're not going to die of blood clots, so that's cool. Yeah. And I'm oversimplifying it. That's not exactly what the study said, but it was one study. It wasn't a very good one. So no, there's not like I'm drinking some vodka every night for my health. That's not a thing. A glass of wine, yes, it contains some antioxidants. So does grape juice. Drink the grape juice. But only 1 glass because it's still high in sugar. So there's still there's healthy things, there's unhealthy things in basically everything. Nutrition is a complex, messy thing, and our understanding of it is pretty dumb, honestly. Yeah, we could definitely get way into that though if you want. Okie doke. All right, and again, that collecting duct is affected by ADH. I think we're good. I think that was about it for the nephron loop. Are you guys feeling? We have not done GFR yet, so if you've got blanks related to GFR, if the juxtaglomerular apparatus, we haven't gotten there yet. Those blanks should be there, but anything else related to the nephron loop should be pretty much filled out. Are you feeling good about your worksheet so far? Any questions from the worksheet?
Renal clearance, are we going to get
Yeah, we haven't gotten to renal clearance yet. That's correct.
What substances are secreted with PCT to make it to the
PCT, what's secreted?
drugs?
PCT is drugs. DCT, potassium and hydrogen.
Oh, I thought there was.
Did I miss the maculadenza?
Nope.
No.
That's part of the juxtaglomerular apparatus. Yeah, we're not there yet. Okay, let's talk about things that act on GFR, glomerular filtration rate. What this means is the rate at which the glomerulus filters. And it's going to be used as a measure for kidney function, which is why it's really important to you. It's going to tell you how healthy your kidneys are. Your kidneys should be filtering at about 125 milliliters per minute. My chart says, 120 is the high end of normal. It should be, actually 125 should be normal, straight up. But if you're over 60 milliliters per minute for your glomerular filtration rate, that's considered within a healthy range. Kidney disease is considered 15 to 60 milliliters per minute. That would be the chronic. And then acute kidney failure would be less than 15 milliliters per minute. So that's quite a bit of lack of function and inability to produce, urine sufficient to get rid of wastes while still retaining water. So a lot of things are going to act on glomerular filtration rate. First of all, net filtration pressure. And this is what we were getting at earlier. It's a capillary bed, therefore your hydrostatic pressure is going to push water out, and your osmotic pressure is going to pull water in, but overall, what tends to be stronger at the arterial end? Is it going to be hydrostatic or osmotic pressure? More powerful.
Oh,
Hydrostatic is always going to be more powerful, especially when you're talking about arteries. Another effect is going to be total surface area available for filtration. That means having 2 kidneys instead of 1. That means having a number of functioning nephrons. It is possible to damage individual nephrons. It also is going to be affected by glomerular cells. There are going to be cells that can contract and decrease surface area. And then filtration membrane permeability. So they are fenestrated, so they should be more permeable than capillary beds with tight junctions, for example. Not as permeable as sinusoidal capillaries, but again, we do not want to lose red blood cells into urine, so that's a good thing. So if you have an elevated GFR, if GFR is high, your urine output will also be high. And when you pee out more, you are dehydrated, in the literal sense of there is less fluid volume in your system, and that decreases your blood pressure. Blood volume decreases, blood pressure decreases. And vice versa, so if GFR is low, urine output is low, and then you retain water, that water is in your bloodstream, your blood pressure is high. Yes?
So how is it like the more water you drink, more pee, but you're not dehydrated?
It's all a balance, right? So the more water you drink, more water you drink, the more hydrated you are, the less ADH you produce, the more you urinate back out. So it's going to be homeostatic. We're going to want to get back into that homeostatic range. If we drink too much, we're going to pee more out. If we drink too little, we're going to pee less out. So we're always going to be balancing in that range. And it's only when we get outside of those healthy ranges, if we're in the desert and we're dehydrating to death, that it gets into a dangerous zone. Yes?
How do they test the GFR?
Many tests for GFR, and we'll talk about some of those that's coming up. Okay, so there are intrinsic controls and extrinsic controls that will act on GFR. First is going to be our intrinsic controls. We're going to talk about how the smooth muscle of blood vessels directly affect GFR. And there's also going to be tubuloglomerular feedback. We're going to have a feedback system between the distal convoluted tubule and the glomerulus. They're actually touching each other. They're going to communicate with each other so that we can alter GFR as needed. So myogenic mechanism first. First and foremost, we've got an afferent and an efferent arteriole, and those do have layers of smooth muscle around them. Okay, so think about hydrostatic pressure. We're letting a certain amount of fluid into the glomerulus. Now let's say we constrict the smooth muscle around the afferent, the arriving arteriole. What happens to the blood volume and blood pressure within the glomerulus if you constrict the afferent arteriole? Decrease. And then what happens to hydrostatic pressure if we decrease the blood volume? It's going to decrease. So constricting the afferent arteriole is going to decrease GFR by decreasing the hydrostatic pressure of the capillary bed. Now imagine we then constrict the efferent arteriole and relax the afferent arteriole. What happens to the blood volume within the glomerulus? It increases. And what happens to hydrostatic pressure? It also increases. And then what happens to GFR? Increases. So literally all that the kidneys have to do in response to blood pressure changes is constrict either the afferent or efferent arteriole. And that's the myogenic mechanism.
Both of them are the myogenic.
Mm-hmm. So what it's doing in response to is changes to blood pressure. Now here's the thing, this capillary bed is delicate. Capillary beds are delicate, that is their nature. Simple squamous epithelium as small as it gets, right? So if you have high blood pressure, let's say you have chronically high blood pressure, that's going to put a lot of stress on that glomerulus. So this is one mechanism that is protective of that capillary bed. Let's constrict the afferent arteriole to prevent the systemic blood pressure from literally just like crashing through our filtration system. Does that work for everyone? It works for the people up front. Does it work for the people in the back? Kind of.
So,
How about people in the middle? Are we at 50-50? Is it like 150 and 0 as we go back? Yes.
so your body changes. So if you have hypertension, your body will just automatically increase that resistance to afferent.
Generally speaking, yes. We are going to have other mechanisms acting on it. There are going to be things that utilize the myogenic mechanism too as feedback, but yes, that is what we're getting at, is that high blood pressure is going to be dangerous, so we're going to cut off that blood pressure to the glomerulus to protect it.
Okay.
Okay, so elevated blood pressure means the muscles stretch, which means in response they constrict that afferent arteriole to restrict blood flow into the glomerulus and protect it from damaging high blood pressure. And the low blood pressure is going to do the opposite in the afferent arteriole. Next you have tubuloglomerular feedback. We took the nephron and we stretched it out into a line, but in real life that line is in 3 dimensions and it wraps around itself, right? So in real life, the distal convoluted tubule is in direct contact with both the afferent and efferent arteriole, so that the concentration of stuff in the DCT can impact the glomerular filtration rate. What's going to happen is it's going to detect, cells of the distal convoluted tubule are going to detect sodium chloride. Now, if your glomerular filtration rate is high, that means your filtrate flow rate is high. Things are getting pushed from behind, so literally if it starts faster, it stays faster. That means there's not a lot of time for stuff to reabsorb. That means there wasn't enough time in the PCT and the loop of Henle for sodium to be reabsorbed into the cardiovascular system and then for water to be reabsorbed. So what that means is that by the time it gets to the distal convoluted tubule, the sodium level is too high. And special cells known as macula densa cells will detect this elevated sodium chloride concentration. And it's going to give that feedback back to the myogenic controls and cause constriction of the afferent arteriole. Absolutely, I can repeat that, yes. I know that requires repeating. big picture, what this series of complicated arrows means is that if your sodium level is too high in the DCT, you did not have enough reabsorption time, and we need to slow it down. Did that make more sense? Okay. So ignore the arrows for just a second. If you have high sodium, there wasn't enough time to reabsorb it, and we got to slow it down.
What would cause that?
So elevated GFR, like high blood pressure or something, or high blood volume or something like that. Yep. Did that make more sense? Okay. So the opposite is also true. If you have low sodium chloride, that means your GFR is really low. And those macula-densa cells will detect the really low sodium levels and try to speed up your GFR. Again, using that myogenic mechanism. So remember, we've got two places we constrict. If the GFR is too high, we'll constrict the afferent, and that'll slow it down. If it's too low, we'll constrict the efferent, and that will speed it up. Very good question.
Sorry. The macula densa is what causes the constriction, or like signals.
The feedback. Yeah, so the macula densa cells are detecting the sodium chloride, and then they're going to communicate with the capillary walls. So that was it for intrinsic controls. That was how the nephron affects its own GFR directly. Next, we're going to start talking about extrinsic controls, the most direct of which would be the sympathetic nervous system. When you have norepinephrine from the sympathetic nervous system and epinephrine from the sympathetic nervous system, those are both released by the adrenal medulla, and that's going to cause systemic vasoconstriction. That's going to increase your blood pressure, and that's going to alter your GFR. You've done the RAS system before, so this should be review for you. Do you remember what releases renin? Nope. The liver will be involved in a second, but not yet. Not the adrenal glands. Your kidneys. Your kidneys release renin. So it is an extrinsic control, but it's still your kidneys. Your kidneys are really involved with blood pressure, right? This is like the key thing here, is your kidneys are going to freak out if your blood pressure is messed up, so they have intrinsic controls to affect the blood pressure within them, and they have extrinsic controls that affects not only the blood pressure for the kidneys, but of everywhere else in the body. So they are very key players in your blood pressure. So you're going to have your kidneys detecting blood pressure changes and releasing renin when your blood pressure is low. Now that renin is going to cause the conversion of angiotensinogen into angiotensin 1. What released that angiotensinogen?
Liver.
The liver. There we go. Now is the time for the liver. And then you have angiotensin converting enzyme that converts it to angiotensin 2. And that causes ADH and aldosterone release as well as thirst. And that makes you drink water and retain water. And that's going to raise your blood volume, and that's going to raise your blood pressure. So it's pretty cool. You've got ways that your kidney affects its own blood pressure just to protect itself, and then it's going to impact your systemic blood pressure via that RAS pathway. And again, RAS is going to be activated, again, by those macula densa cells in response to sodium chloride concentration. Same way that they had feedback with the myogenic. Let me see if there's something else I need to tell you about this. All right, I believe you have the word granular cells on your worksheet. Granular cells of the juxtaglomerular apparatus are the ones that release the renin. The macula densa cells were the ones that sense sodium chloride concentration.
So are granular cells only found in the kidney since they release?
Yes, these granular cells. Sometimes we'll use the word granular to refer to a variety of appearances of different cells. So you have like stratum granulosum in your skin, for example.
So how do we find them?
Juxtaglomerular apparatus is pictured here. There's actually other slides that have images of them. Let's go track one of those down. I know you can't see it for a second. I'm going to go find a picture. The juxtaglomerular apparatus will be defined as the macula densa of the distal convoluted tubule and its contact with the afferent and efferent arterioles. So this is also an image of the JGA.
So is it like, what was it, the second part?
So you have the macula densa cells of the DCT, the granular cells of the afferent arteriole that are going to release that, renin, and help with that myogenic feedback system, also extraglomerular mesangial cells, and their contact with the afferent and efferent arteriole. So this whole thing is the apparatus right here.
So is it basically the system that runs the macula densa and the granular cells?
Yeah. I mean, it's those cells working together. Those are the cells that work together, the JGA.
And that's where they share the basement membrane?
Mm-hmm. So be careful, the basement membrane you may be thinking about. Are you thinking about the filtration membrane over here? Okay. So filtration membrane has the shared basement membrane. These are just in contact.
Okay.
So don't worry about basement membrane for that. And I'm sure if you have that question, others have that question. So thank you for bringing it up. Okay. Yes. How are you guys feeling about nephrons? How are you guys feeling about juxtaglomerular apparatuses? How are you feeling about intrinsic controls of blood pressure? How are you feeling about extrinsic controls of blood pressure? I got one thumbs up because you've seen RAS before. Okay. Remember, it's only 9:30 in the morning and we've got till 4 p.m. to get this. So.
I'm just confused because it sounded like those mechanisms were both ways. Do they all work both ways?
Do you mean to both increase or decrease blood pressure? Yes, they work both ways in response to homeostatic mechanisms. So if it's too high, they'll make it lower. If it's too low, they'll make it higher. So if your blood pressure is already high, your kidneys should stop releasing renin, is how that would work. Does that make sense? Okay. Yeah, thanks for bringing that up too. Yeah. So what we mean by renal clearance is how much of a drug is cleared by the kidney. So drugs leave the system in a variety of ways. They can be broken down by the liver, but usually at some point we're going to have to excrete them. So we could do it through urine, through feces, depending on the individual drug. And one thing that you may be catching on to with this whole renal system is that we've got this complex system of reabsorption and secretion happening where not everything that makes it to the tubules is going to leave your body. So clearance times, clearance percentages are going to vary. Now some of the terminology here is a little bit weird. We've got renal clearance rate, and it's going to be related to the concentration of the substance, the flow rate of urine formation, sort of your GFR, and it's going to be inversely related to the concentration of same substance in the plasma versus in the tubule system. Yes?
So basically, renal clearance is the concentration and the rate with which stuff gets out of urine?
Yeah, the rate at which stuff leaves through urine, leaves your system through urine. Now, there's a sort of standard. We use inulin as one way that we detect GFR. Inulin is a plant polysaccharide. That means it's a sugar chain that we can't break down. And it's standard used because it is neither reabsorbed nor secreted. And we are using these official definitions for reabsorbed nor secreted. It is filtered. It goes from the glomerulus to the filtrate. But from there on out, throughout the entire tubule system, it will not be secreted nor reabsorbed. So once it's in the tubule system as filtrate, all of it is going to be excreted as urine. And that's going to be one of our tests for GFR. We're going to use that clearance rate of 100%. And if we filter 125 milliliters per minute worth of inulin, then our GFR is 125. Okay. So that's, again, what we mean here is inulin will be filtered, but it's not going to go back to the peritubular capillaries, and that it's not going to go from peritubular capillaries back to here. So not all of the inulin makes it out into urine all at once, right? Some of it is filtered, some of it remains in capillary beds, but none of it crosses over. So far so good on that? It's a weird concept. It took me a few semesters to really get this concept, so it's okay if you're a little stumped right now.
Is that like special for inulin?
It is special for inulin. Other drugs will have different clearance rates. We're bringing up inulin specifically because it's a good measure for GFR. And that's really all you need to know about that. When you get to pharm, there's going to be a lot more about, half-lives of drugs and clearance rates and things like that. All we're doing is the big picture right now of saying drugs will be secreted and reabsorbed at different places. They'll have different clearance rates. Inulin happens to be the one that is 100% secreted after it's filtered. Or sorry, 100% clearance rate. Neither secreted nor reabsorbed through the tubule system. Okay, big picture questions about that? It does come out in the urine. So some of it is filtered, but not all of it leaves in filtrate, right? So you have a certain amount, and some of it goes to filtrate, some of it just didn't get out into the filtrate, right? But everything that made it into filtrate is going to be peed out. It's all gone. Yeah, not all of it went in. So like imagine, I understand what you're getting at. I totally understand where this is coming from, and I think it's totally valid that you're bringing it up. Imagine you've got a big bucket, and you've got a little Brita water filter pitcher over here. You're going to pour that bucket of water right over that Brita water filter. Everything that makes it into the Brita water filter is going to go through the Brita water filter, but a bunch of that water is going to splash to the sides. None of the water that splashed to the sides is going to make it into the Brita water filter. None of the Brita water is going to make it outside of the Brita water filter. So that 100% clearance rate was all of it that made it into the Brita water filter goes through the Brita water filter. I don't think it really does much. I don't really know what the role of inulin is. It's a plant polysaccharide, which means we're not breaking it down. Yeah, we can look up to see if there's a function of inulin as a drug, but I have no idea. I'm going to Google this now. I'm going to pause and Google. So for the recording, we looked up inulin, and it is basically dietary fiber, is what we're getting at. Lots of plants contain inulin naturally. You can buy inulin as a nutritional support. Within plants use it for energy and for resistance to cold, but humans use it for dietary fiber. All right, let's wrap this up. We've got a little bit more. We talked about chronic renal disease, renal failure. We didn't really talk about nitrogen and proteins that much. We mentioned that at the proximal convoluted tubule, you are going to reabsorb some amino acids, and we kind of just skimmed over the fact that the whole purpose of this is getting rid of nitrogenous wastes, which means protein breakdown. You may not understand that proteins contain nitrogen in their carbon-based backbone until you get to chemistry or beyond. Today we're just going to roll with that concept. Nitrogen means protein. And uremia. Urea is a nitrogenous waste. Ammonia is a breakdown of that same nitrogenous waste. So if your kidneys fail, you're going to have a buildup of nitrogenous wastes, and we're going to call that uremia. And that nitrogenous waste floating around in your circulatory system and making it throughout your body, including to your brain, can cause all kinds of problems such as dementia, toxic molecule accumulation, and ionic and hormonal imbalances will also be part of renal failure. So we can do hemodialysis, we can do kidney transplants. If you're familiar with hemodialysis, it's going to be very, very time-consuming and very, very life-altering, and it's not going to be a permanent solution. It's going to be a very short-term solution. Now, normal color, urine should be clear and pale yellow. If it's cloudy, that might mean there's urinary tract infection. That probably means there's white blood cells in it. If it's darker yellow, that means you're dehydrated for the most part. If it's pink, brown, or smoky, it could be from the foods you ate. It could be something not harmful at all. It could be beets. It's okay. Bile pigments, blood, or drugs. Some drugs will alter the color of urine considerably. Yes?
What's the difference between cloudy and smoky?
Smoky's going to be brown, yeah, whereas cloudy's going to be white. Your urine should not normally smell terrible. Your urine should only be slightly aromatic. The smell of ammonia, the reason your cat's litter box smells so bad is because the bacteria metabolizes things, and that urea is actually broken down into ammonia. So that's the ammonia smell you get from the cat's litter box. It's because it's sitting there for a long time. Yes?
Why does food asparagus make it smell?
It's just the metabolism of certain things that are in asparagus, and this is actually my next point. Raise your hand if you don't know what asparagus pea smells like. Does anybody not know what asparagus pea smells like? Everybody in here knows what asparagus pea smells like? Because there's a genetic mutation where some people can't smell the molecule that is broken down from asparagus that is found in urine. So every once in a while I get a couple of people raising their hand and saying, I have no idea what asparagus pea smells like. And you're like, well, you're living in blissful ignorance because you've been in a cloud of asparagus pea before and you just don't know it. It's harmless. It's a harmless mutation. It doesn't hurt anybody. You just get to live in blissful ignorance of asparagus pea smell. That's all. Okay. And at that point, usually people ask, well, then what does asparagus pea smell like? And everybody goes, oh, it's so hard to explain. And that's the way that word goes. All right, urine should be slightly acidic. That's a generally good thing because that's going to help bacteria that are crawling up your urethra right now. And specific gravity is about concentration, so it should be about that concentrated. That's all I got for you. Have you guys heard of an alkaline diet and sort of the health food fad? Oh, I'm drinking all the alkaline water now because it's going to help me and be healthy. And I think if you've gotten nothing else from today, just imagine how ineffective that alkaline water is going to be in actually changing your health or doing anything good. All right, we talked about nitrogenous wastes, urea. I'm just proving to you over here that urea does in fact have nitrogen in it. That is why we call it a nitrogenous waste. Nitrogen, nitrogen, right there. You don't have to go and memorize this molecule right now. It's not what it's there for. So in order to detect GFR, one, we can use inulin. We can also use blood urea nitrogen to see how much nitrogenous waste is left in your cardiovascular system. And that's going to be a decent measure of your GFR and your kidney function, right? Going to be a slight problem with that because anytime you eat protein, you are going to have more nitrogen in your system. So it could be that their BUN is elevated and that they have kidney failure, or it could be that they had a steak for dinner and it was delicious. Either one of those things. Same with creatinine. Creatinine is a metabolite of creatine phosphate. Who's heard of creatine phosphate before? We have a similar issue with creatine phosphate. What do we use creatine phosphate for? Working out. Bodybuilders will use creatine phosphate as basically an energy source. It's actually going to donate that phosphate group to ADP and help us make more ATP for more cellular energy. So it is actually beneficial for that cellular respiration pathway. And yeah, it's absolutely going to increase cellular energy. So your creatine could be elevated because your kidneys are failing, or it could be elevated because you took some supplements and worked out hard yesterday. Which is why we're going to use so many different measures for GFR. We're not going to use just one, because outside things are going to influence them. You should have a certain amount of sodium, potassium, phosphate, sulfate, calcium, magnesium, and I think this is this bicarb or is this hydrochloric acid? I think it's hydrochloric acid. I'm going to go with HCl. Yeah. No, because it would have a Cl in there. Oh well, we'll figure it out. And again, you do not want blood, elevated proteins, elevated white blood cells, or elevated bile pigments in urine.